Mute myself. Yes, thank you very much. And thank you for inviting me to come and speak. So I'm going to talk about sort of one of my chapters of my DPhil, which we call PhDs at Oxford for some reason. And yes, we're as a lab, whoops, we are super interested in understanding the development of the teleost vertebral column. And traditionally, the vertebral column of teleosts has been divided into two domains: the precaudal domain, defined by the presence of pleural ribs, so very, very functionally similar to thoracic vertebrae in tetrapods. They just essentially protect the viscera. And caudal vertebrae, these sort of tail vertebrae which lack pleural ribs and obviously are really important in the structure of the tail. But what's really cool from a developmental perspective is that we have these adult phenotypes, the total number of vertebrae, and the type of vertebrae, which can act as proxies for these developmental mechanisms of somatogenesis and regionalization. And so somatogenesis is this kind of cyclical generation of somites, and then these somites go on to form vertebral precursors. And so we have a zebrafish embryo here, and we can see that as the tail, this is the tail, as it extends sort of posteriorly, we get these budding of somites from this pre-Semitic mesoderm. This is a highly regulated process, and obviously it is common to all vertebrate animals. And so this very, very cool developmental process. So ultimately, the number of somites roughly determines the number of vertebrae in the teleost vertebral column. And so resegmentation actually doesn't happen in teleosts. So we have a very, very nice relationship between the number of somites and the total number of vertebrae. Similarly, Hox patterning patterns the anterior-posterior axis, providing identity for these somites to adopt, which enables them, it specifies these kind of different boundaries along the vertebral column. So Hox genes are probably really important in patterning this precaudal boundary, for example. And so we have this really cool adult phenotype that sort of it's almost directly linkable to development. And so this is really interesting from a macroevolutionary perspective, because we can use vertebral counts and proportions to tell us something about how somatogenesis and regionalization have evolved as animals have diversified. And so I'm really fascinated by this amazing group of animals called African cichlids, which are, of course, endemic or found in the natural fresh waterways of Africa. And they're extremely diverse. And there's lots and lots of tribes. There are hundreds and hundreds of species. But what you'll see very quickly is that they have very, very different body shapes. And that also means very different vertebral counts and vertebral proportions. Most of the species diversity is contained within the Great Lakes of the East African Rift Valley. So these are Lake Victoria, Lake Tanganyika, and Lake Malawi. And so actually Lake Malawi are the species that we're most interested in the lab. And really, they've undergone these absolutely massive adaptive radiations. So over the last sort of 800,000 years in Lake Malawi, from one single common ancestor, about 850 species have evolved, which is utterly remarkable. And they're more genetically closely related than human populations are. So for that reason alone, and the diversity, and also the fact that they're very, very well phylogenetically resolved, they offer this really excellent model for macroevolutionary studies. And so my data collection was very, very simple. Really, all I've done is landmarked and collect data from 4,544 specimens. So just 2D lateral X-rays, which boils down to about 569 species, which actually then, if we look at how many are retained on the phylogeny, that's about 429. And literally all I've done is count manually, I should add, count the number of precaudal vertebrae and caudal vertebrae. And also I've now added some body aspect ratio landmarks. And in fact, now, this is a slightly old slide, I've added an aspect ratio for sort of the cranium, for the head region, and for the tail region too. So we have these very, very simple kind of linear and counting measures to look at kind of how the axiomorph space has evolved and see what we can infer about somatogenesis and regionalization as African cichlids have diversified. So the first thing that we notice, so we threw kind of all of these measures into a phylogenetic principal component analysis. We find that actually PC1 is kind of, as you go more positive, it's an increasing overall elongation. So you're lots of vertebrae, you're very elongate, so you're longer in you're longer and slimmer. And PC2 turns out to be sort of a regionalization type problem. So it's just increasing caudal domain, so this tail region. And actually what we see, and it's quite a nice sort of relationship, is actually that the lake species occupy this kind of top right part of this axiomorphal space, which suggests that actually these lake species are more elongated than riverine sort of counterparts. Your riverines are down here, the lake species are up here. They're probably doing this by elongating their caudal domains. So they're having these very, very relatively long tail sort of regions. And actually we see this really nice trend where the kind of expiration of this sort of axiomorphal space sort of correlates with the age of these respective sort of lake radiations. So Lake Victoria is very young. It's about less than 100,000 years old. Lake Malawi, again, is about 800,000 years old. And Lake Tanganyika is about 12 million years old. So it's almost like you can use Lake Victoria and Lake Malawi to sort of, and Lake Tanganyika is sort of like this future progression, as if all of these sort of remain stable, you're going to almost sort of replicate this kind of Lake Tanganyikan disparity in axiomorphal space. What's really cool is, so two things are going on. So how are you elongating your body? So elongation seems to be really important. One way that you're doing that is by adding vertebrae. So adding vertebrae is elongating the body. So something is going on with symmetrogenesis in that regard, but actually not much. So when you correct for phylogeny, only about 25% of your variation in the body aspect ratio is being explained by the addition of vertebrae. So clearly adding vertebrae is very important, but it's not the only thing that's important in elongating yourself. Nonetheless, the species that tend to be elongating themselves tend to fall into two categories, or both, being pelagic, so being adapted to very deep water, and being piscivorous, so being adapted to hunting and eating other fish. I should preface this by saying actually specifically it's being adapted to open water hunting. You're being an active predator, because actually there are a lot of piscivores down here which are actually ambush predators that haven't elongated themselves. So this is a very unique sort of driving factor for elongation. But nonetheless, the addition of vertebrae seems to be important in elongating yourself. But so something is going on in terms of modifying symmetrogenesis, and it's important that you have to do that in order to adapt to these different ecological niches. But what's really interesting is that intraspecific variability in the number of vertebrae is very low within a species. So actually there is variation within a species about how the number of vertebrae that's formed. And actually, if we look at the whole African phylogeny, this hasn't really changed as African cichlids have diversified. And so this variability we're kind of reading as a sort of Semitic fidelity. How good is this process of symmetrogenesis of generating the same number of somites each and every time within a species? And so actually where you see these lighter colors, it's probably mislabeled species, because there's a lot of, the taxonomy is very messy. But the intraspecific variation hasn't changed, which is strange because you're elongating different lineages, you're adding and removing vertebrae. So why hasn't this variation changed? And we were interested in looking at whether intraspecific variation sort of correlates with vertebral counts, because we had this idea, well, some lineages, because they've diversified very quickly, maybe they've retained some sort of Semitic fidelity, this variation in the number of vertebrae, which they've been able to act on, and they just haven't selected it out. And so we might see a kind of a positive selection signature. We don't see that. But then we thought, or maybe some lineages have done this, they've taken a very long time, so maybe we'll see negative selection, or actually the variation decreases with time because actually you want to select out the individuals within your population that are poorly adapted. We don't actually see any relationship between the total count and the intraspecific variation. So this really suggests that symmetrogenesis itself as sort of a mechanism is probably not what is being selected for here, even though the number of vertebrae are changing. So then we thought, well, actually, maybe just adding one vertebrae is enough to kind of push you over. And because obviously the thing that natural selection sees, for want of a better phrase, is your body aspect ratio. And so we thought, well, maybe there's a little difference. It turns out, so this species is a potential sympatric ancestor for all Lake Malawi cichlids. So it's actually a very good candidate to look at this kind of question in. But actually, we see that there's very little difference in the body aspect ratio in terms of the number of vertebrae that you have. So it's slightly mysterious as to where this is, how you're able to change the number of vertebrae without changing this sort of Semitic fidelity. But it leaves us really with two kind of hypotheses. It's potentially the coevolution of other traits that also happens to alter the somite/vertebral count, which is entirely possible. And a slightly related question is, well, you're probably potentially modifying development prior to semitogenesis. And so we have a few hypotheses, or in fact, we have more than hypotheses now. We have a lot of data to kind of support that hypothesis. And finally, I sort of want to talk about some kind of...
Callum, can I just interrupt? You've got 1 more minute.
Okay, that's fine.
Thank you.
This is my last slide anyway. So we wanted to look at how semitogenesis and regionalization are related. And actually, we find that basically semitogenesis, Semitic changes in the vertebral column are usually accompanied by a co-occurring homeotic shift, essentially, which has some interesting developmental implications. So riverine and lake cichlids are being driven to adopt different axial morphologies, and elongate lineages tend to be pelagic and or be piscivorous. Despite vertebral counts being evolvable, intraspecific variation is very low, and changes in total count are usually accompanied by a shift in regionalization. And we're trying to look at how that fits into our current AP patterning model. So I'd just like to thank everyone, particularly my supervisor, Dr. Berta Verd, and Professor Roger Benson. And obviously it takes a village to train a PhD student. So yeah, thank you.
Thank you so much. Thank you very much, Callum. We've got time for one quick question. And so I'm just going to read this one out. Thank you for your talk. Can you comment more regarding the mechanism of increasing vertebrae number? Does semitogenesis happen more quickly relative to organismal development, so more vertebrae are made during semitogenesis, or is the semitogenesis period extended?
So we actually think the answer is that semitogenesis is extended in these elongate lineages. So there's two things going on. Semitogenesis is extended, and actually the pre-Semitic mesoderm that you have available to segment is probably larger. And so we're actually thinking that you just start out initially, even before gastrulation or even as gastrulation is happening, with a bigger embryo, which means that you have more tissue to segment once gastrulation is actually finished, which might be a very, very quick way in terms of elongating your vertebral column.